Hello and welcome to this introduction to Workspaces. Workspaces can be found in the Workspaces section in the sidebar. There are a way to upload your data and examine it and explore it without attaching it to a particular study. So if you have some CSVs with observational measurements or with modeling predictions, you can upload them and visualize them and even fit some basic growth models to them. We can see that this section, the upload data section, includes a direct upload subsection where we'll see that the data will show up, and an API data subsection where we can push data using the programmatic interface. This will be covered in a separate video. For now, we're going to focus on direct uploading. The right-hand side shows a form where we can upload our CSVs. We can see a preview of what the format that is expected. The first column would be interpreted as time, and the other ones will be interpreted as measurement values. Now, if we were to click on the include error columns checkbox, the first column is still time, and then the columns are going to be paired a value, and then an error column, a value column, and then an error column. Just so you can see how this looks, we can use the example data that is shown here. I've already downloaded this. I'm going to click on browse and pick this particular CSV file, and we'll see that the headers are parsed as the time column, as this FC live, etc. column. This is interpreted as a value, and this STD column is also interpreted as a value, which means we should click on include error columns to interpret it as a standard deviation. The metadata is optional. We don't have to upload it, but if we do, we can compare and convert units between different CSVs, so it's highly recommended. We can say right now that this is observational measurements. This is flow cytometry measurements of live cells in a particular community, in a particular study that I've downloaded. This is a measurement of a microbial strain, which means the units that are shown here are going to be growth units. If it were a metabolite, we would see metabolite specific units. And let's say these are cells per milliliter. We can upload them, and then we see them show up here in the direct upload section. We see the metadata that we entered, measurement, strain, and so on, and a timestamp of when it was uploaded. We can edit this record to change the label, for example, of BHI monoculture or something like this. So it could be we could change it in some way that is more consistent with the rest of the data that we upload, for example, and then click on save to update this. Now I'm going to do the same with this measurement here, which also includes an error column, and I'm going to again pick observational measurements of a strain in the same units, and I'm going to add this model here, which it doesn't matter at this point if we pick error columns or not, because there's only 2 columns, but this is a model prediction of a strain, and we can see now that all 3 of these all 3 of these example files have been uploaded successfully. At this point, we can go to the Visualize tab to try to explore this data. I can pick, for example, these 2 data traces, one that represents an observational measurement and one that represents a model of these observational measurements. And I can compare it with the data from other studies. For example, I can go to the compare data source, and these data traces have been collected by me using the compare interface that you can find in other places on the web application. And for example, I can say, well, bacteria at the Omicron we have here in one experiment. We could perhaps compare it with this one, and we'll see that this looks quite odd. The data does not compare at all. If I go to log view, I can understand what the problem is. The problem seems to be that there's a mismatch in the units. And this is one way for me to validate that the data I uploaded here is actually not in cells per milliliter. It is in cells per microliter. And I can edit these. And let's see, edit these as well. And now if I go back here, and I do the same thing. I compare these with these flow cytometry values. We can see that they are much better, more comparable, much closer to reality. We can use the buttons here on each of these individual data traces to download the data, download the individual data traces as a CSV file. We can click on the Visualize button to directly jump to the Visualize tab with this particular data trace ready to visualize. We can, as you saw, edit these individual traces, or we can also delete them. If we look at the right-hand side where the upload form is located, there's a number of other controls. One is to completely clear the data here. Workspaces are meant to be somewhat temporary. Ideally, you should upload studies with rich metadata. But again, the point here is to simply export the data and maybe share with other people. In order to do this, one thing that can be useful is to create different workspaces. On the top side, you can see that this is a default workspace. Every user has one, but we can create a, let's say, share or like a lab workspace or something like this to send it to other people. Let's say create this. And now I'm at a brand new workspace and I can go back to the default and see all this data. Or I can go here and pick some kind of other example here. Maybe I can upload it like this. Right, and now I can maybe share this particular page with other people. However, note the message here that says this page is private. If we try to open this into a private browser, we'll see that we will not be able to see this. As you can see, if we open this in a private browser so that we are not logged in, we see a 403 forbidden page. We're not able to access it. However, what we could do is click on the publish button, and in this case, the state of the page changes. This page is visible to anyone with the link, and in fact, if we refresh this page, we can see this particular data upload. We can click on Visualize and visualize this data here in the browser. Note that there are no units here because I did not provide any metadata. Again, this is perfectly allowed. If you don't provide any metadata, you'll just be able to see the visualization as it is, and you can add the metadata later if you choose to. One thing that we can do is we can upload a data file with many more columns. For example, here I uploaded a file that has a value column with OD values, with pH values, glucose, pyruvate, and so on. So that's quite a lot of columns. They do not have SCD columns. If I click on SCD, or they do not have error columns. So if you click on this, this will assume that every other one is an error column. So I better be careful about this. I cannot really say that this is a measurement of a community restraint or anything like this because it's a mixed one. But I can say it's observational measurements. Because most of these are metabolites, maybe I can say this is a metabolite in millimolars. You can see MM here, and then edit the metadata for the others to fix this. Either way, if I click on upload, you can see that all of these get uploaded, and I can edit these two to say that this is the observational measurements of a community, and it is in unitless growth units, so it's an OD. And this is something else. This is a pH value. And so if I now go to the Visualize panel, you can see all of these are available. We can select them here to add them in the same place. And by doing this, we can, for example, remove the pH and move the OD values and the pH and the metabolites on separate axes, for example. And this can potentially allow us to have to upload batches of different measurements. And if we mess something up, if we want to re-upload, for example, replace them, we can always clear the workspace uploads or even delete the workspace and create a brand new one. We can also fit some basic growth models to the data that we've uploaded. For instance, I can pick this one and choose the easy linear method and click on calculate. and give it a little bit of time to see this tangent method, this easy linear method that determines the growth rate, the observed growth rate based on regressing between 5 points. Or I can pick the Roberts model and pick an end time around here, for example 40, to calculate a growth curve in that way. And if I go back to visualize, both of these models are going to be available here under this field, and they will also be available here to download predictions of these fits if we need to use them in other kinds of software. That's all about Workspaces for now. If you have any suggestions about what could be useful to you in this kind of functionality, feel free to go to the About Us page and find our contacts and reach out and discuss. Thank you very much for watching, and good luck with your research.